Hello, and welcome to our webinar, How to Optimize CRISPR Genome Editing in Primary T Cells. My name is Molly Schubert, and I'm a product manager in the Genomics Medicine and CRISPR group at IDT. I will be serving as the moderator for today's presentation. Today's webinar will be presented by Bernice Demandrew. Bernice is a research scientist in the Molecular Genetics Research Group at IDT. She received her master's in molecular physiology and biophysics from the University of Iowa, where she studied transcriptional regulation of multi-drug resistance in pathogenic fungi of the lung. At IDT, Bernice has focused on optimizing delivery strategies for CRISPR reagents, as well as methods for increasing the rate of homology-directed repair. During today's webinar, please submit your questions using the question box. Any questions that are not answered during the Q&A session will be followed up with afterward.
Thank you very much, Molly, for that introduction. My name is Bernice Demandrew. I've been at IDT in the CRISPR R&D group for about 6.5 years, so quite a bit of time at IDT. In the last couple years, I've worked on this project optimizing CRISPR editing in primary T cells, and so today I'm going to talk to you about some of the optimization work and what led us to improve rates of editing in primary T cells. And here's an overview of what I'll talk about today. We'll start with a bit of background on CRISPR editing and what it's used for. Then we'll talk about optimizing RNP delivery in these cells, starting with the cell culture itself and some electroporation parameters. Next, we'll talk about methods to improve HDR with various enhancers. We'll talk about short inserts and making SNPs and large knock-in. And we'll wrap up the presentation with conclusions, and finally we'll take questions from the audience and answer as many as we can with the time we have. So CRISPR at its core is a method to target genomic DNA. And with wild-type Cas9 nuclease, there's a targeted intended double-strand break. But the interesting part of CRISPR comes in the repair of DNA, and that's where different reagents can impact the repair outcome. So following a double-strand break, there are a few main repair pathways that are important, and mainly today I'll talk about non-homologous end joining, or NHEJ, which give rise to random indels, and this is most useful for making a knockout via nonsense-mediated decay of a protein. Another pathway is homology-directed repair, or HDR, and this is when an exogenous donor template can be provided along with the Cas9 nuclease, and the repair machinery in the cell can use that template to repair the double-strand break and can bring along with it a desired insert or SNP change. Okay, so now that we have an idea of what CRISPR is and what it can do, we can talk about how to do CRISPR specifically in primary T lymphocytes. So in order to efficiently utilize CRISPR in primary T, thorough optimization of primary T cell culture is a really key variable. So T cells should be activated and be actively dividing before editing, which is especially important for HDR, so we'll talk about that a bit later. And in this experiment, we've compared different activation methods that are commonly used in the T cell field, and then always comparing back to an unstimulated control. And we measured CRISPR editing after different time points of activation, so after 24, 48, and 72 hours. So our control here is the unstimulated. Then we looked at use of IL-2 alone. One method is to add CD3 and CD28 directly to the growth media. We also used a commercial activator from stem cell technologies called Immunoccult. We used another mitogen that's called concanavalin A. We tested standard dynabeads, and then plate-bound activation where the CD3 and 28 are in a monolayer on the base of the cell culture dish, and we tested those at low dose and high dose, measuring CRISPR editing after activation for these different time points. And overall, we observed that there are many sufficient methods of activation, but there is certainly a trend that activation for 72 hours is too long. We did not see optimal editing when activated for 72 hours. So 24 to 48 hours is what we found to be optimal for CRISPR editing. That, of course, could change in the primary T cells that you have, and so we recommend doing a base experiment such as this one to observe what is the best activation method and time period for your cells. And next, we optimize a few different parameters for electroporation. Our preferred method is Lonza nucleofection, and for all our RNP delivery experiments, IDT has an electroporation enhancer product. And on the left side of this slide, we're testing 5 different human donors of primary T cells and saw that they all benefited from including electroporation enhancer in the reaction. And on the right, we're testing different nucleofection codes that were identified either from the literature or from Lonza's recommendations. And then we scored for both total editing and cell viability. And we determined which nucleofection code was optimal through this experiment. So going forward, for all the experiments, we include electroporation enhancer and use our top Lonza nucleofection code. Another important factor of T-cell editing is the donor of the cells themselves, and we've seen differences from donor to donor in the ability to edit, like in this graph here, where we've delivered RNP to 7 different genomic loci in 3 unique human donors. And we see a trend that the first donor, which is the blue bars, always edits pretty well across these 7 different loci. Then the second donor in red edits slightly less efficiently, and the third donor in green, even less total editing is observed. So going forward, we always screen multiple donors so that we have more than one biological replicate for each experiment. And the last part of this section is how we determine what dose of RNP to deliver to the T cells via electroporation, or specifically what dose of RNP gives the highest editing, but also where we're not delivering excessive nuclease that can lead to unintended off-target effects in the cell. And we looked at that with different dose conditions of our electroporation enhancer, which I mentioned earlier. So on the left-hand side of this slide, this is the Cas9 dose curve, where when using the electroporation enhancer at various concentrations, we see editing plateaus around 2 micromolar RNP. And then without the electroporation enhancer, we had to go up to 8 micromolar or possibly even higher to achieve decent editing rates. And on the right-hand side is a similar dose curve, but this time with our Cas12a nuclease. And we see much less dependence on the electroporation enhancer with this nuclease, as editing here plateaus around 2 micromolars for all the conditions with and without electroporation enhancer. And so all of that was the important pre-work that was required to establish reliable editing efficiency in primary T cells, which we now consistently see at 80% editing efficiency and 80% cell viability with electroporation. So some conclusions from this section: CRISPR can be used for knockout and knock-in, and CRISPR in primary T cells has a specific, unique set of challenges that have to be optimized at various different steps. Many activation methods will work just fine for knockout, but 24 to 48 hours is optimal for the different activation methods that we tested. And lastly, including the electroporation enhancer increases the efficiency of RNP editing, and around 2 to 4 micromolar is optimal for both Cas9 and Cas12a. Okay, so next I'll talk about how to achieve good rates of HDR in primary T cells. So looking back at this figure, we talked about different repair pathways and how researchers can take advantage of these pathways to get the outcome that we desire. So now I'll turn the discussion more towards HDR, where an exogenous donor template is provided that the cellular repair machinery can use to integrate at the double-strand break site and then can bring a desired insert along with it. So to get us all on the same page for HDR, this little cartoon here is a graphic that outlines the steps that one would take to create a knock-in. So you'd start with identifying your target, and designing an HDR donor that contains homologous sequence that flanks the Cas9 cleavage site, and the desired insert is in the middle. And HDR donors can be viral, double-stranded DNA, single-stranded, plasmid, mini-circles, lots of different options here. And today I'll mostly focus on double-strand DNA donors. The next step is to deliver Cas9 to create a double-strand break in the gene of interest and allow the HDR donor to homologously recombine and bring that insert into the genome. And then the last step is analysis, which can be done lots of different ways, obviously, like flow or sequencing. We tend to do a lot of sequencing with Illumina and Oxford Nanopore platforms. And for small insertions or SNP introduction using single-stranded DNA donors like the IDT donor oligos, there are several references in the literature that can help create guidelines for this type of HDR, including IDT's own HDR methods paper. So we thoroughly investigated different steps of the HDR pathway, starting with guide RNA placement, different modifications on the oligos, targeting versus non-targeting, and all of this information that we gathered here was used to develop our HDR design tool, which is a free tool on our website that can aid in donor design and guide RNA selection. And so we've gathered a lot of tips and tricks and various methods for increasing the rate of HDR. One of them is to use a small molecule enhancer that, when added to the cells post-transfection, can suppress recruitment of NHEJ machinery and therefore increase the rate of repair through HDR. So here we have a collection of different small molecules that we tested in GERCAT cells, and this screen is what led to our commercial product that we call ALT-R HDR Enhancer version 2, and that gave us the greatest rate of HDR when compared to untreated cells and all these other handful of small molecules. And for HDR in primary T cells, we found that it's important to not overload the cells with either donor or the various enhancers in order to maintain the health of the cells for the best HDR rates. So in this experiment, we've varied the concentration of the single-stranded donor, so that's the x-axis here, and then subjected the cells to different combinations of either the electroporation enhancer and the HDR enhancer. So remember that these are two different products. The blue here represents using neither enhancer, red is electroporation enhancer only, green is HDR enhancer only, and the purple is combining both the electroporation enhancer and the HDR enhancer. And so for primary T, we observed that just using the HDR enhancer was sufficient for achieving good HDR rates, and that's likely due to there being a good balance for when the cells are healthy and not overloaded with reagents. And that's where HDR is most efficient. And I mentioned that the mechanism of the HDR enhancer is to block NHEJ, and that's shown in this little diagram on the left side of the slide. We also have in development an alternative mRNA or peptide-based enhancer that we're calling UBVA, and that inhibits an inhibitor of the HDR pathway, so it increases the rate of HDR without interfering with NHEJ. And this is a water-soluble reagent that's delivered to the cells along with the CRISPR HDR reagent, so at the time of transfection in the electroporation reaction, and that just makes it more inert to the cells as they recover and repair after transfection. And furthermore, we've observed that combining UBVA and the small molecule HDR Enhancer version 2 has a synergistic effect. And when combining these two reagents at appropriate doses, that's where you see the highest rates of HDR. And we tested that in primary T cells with two different biological replicates and saw the same effect here. And so what I've shown so far has all been short HDR insertions using single-stranded donors like our IDT ultramers. But what about for larger insertions like a GFP expression cassette or like a CAR knock-in? When we started to investigate large knock-in, we started thinking about what we wanted to achieve and what attributes of a donor were most important for us going forward. So in this table, the attributes that we determined to be the most critical were number one, HDR efficiency, toxicity, cost and turnaround time, and the risk of off-target integration, which will be especially important as the FDA rolls out more requirements for safe genome editing. And so knock-in protocols with viral vectors that insert via random integration and don't allow for precise editing opens you up to a whole host of insertional mutagenesis and off-target effects, so we rule those out right away. Plasmid donors are known to be low cost, but are generally not very efficient and can be extremely toxic to the cells, even in low doses. Linear single-strand donors can be really high efficiency, but are quite difficult to manufacture, which increases both cost to the customer and increased time from order to receiving. And linear double-strand donors are high efficiency and low cost, but they do have a moderate risk of off-target integration by NHEJ. And so this is what led IDT to develop the Alt-R HDR donor block, which is a linear double-strand DNA template with end modifications to decrease the risk of insertion by NHEJ or blunt insertion with off-targeting effects. And like many labs, we started doing our initial screening in immortalized cell lines. So on the left-hand side of the slide, we're comparing different large HDR donor formats in K562 cells. And this is a GFP knock-in at 2 different targets measured by flow cytometry. And again, we saw really low efficiency with plasmid, a moderate increase when using single-stranded DNA and unmodified double-stranded DNA, and then the highest rates of knock-in were achieved with the Alt-R HDR donor block. And on the right-hand side, we also saw the donor block outperform plasmids in primary T cells. And once we established the optimal format of the HDR donor, we looked at other design factors to improve the rate of HDR, like the homology arm length in conjunction with the size of the insert. And so the graph here shows that generally longer homology arms is better, but there is a balance between HDR and the total amount of DNA in the reaction. And so with the 500 base pair insert, there was a point of diminished returns with homology arm length. And now we generally recommend 200 base pair homology arms as a good starting point for any size insert with a large knock-in with the donor blocks. And another design rule that we saw with single-stranded donors for small insertions, which is much more thoroughly described in our methods paper, is the inclusion of mutations in the donor sequence. So mutations in both the protospacer adjacent motif, the PAM, or in the seed or the guide RNA binding sequence can increase HDR efficiency, and that happens by preventing the Cas9 RMP from binding the double-strand donor itself. And including mutations along the repair track will prevent Cas9 from recleaving after the insertion has been made. And this is especially important when PAM availability in your region of interest limits the design choices so that you're not inserting directly at the cut site. And if the insertion is being made further away from the cleavage site, this is where those PAM or track mutations is the best method to get efficient HDR insertion. And so this slide is my last bit of data here, and this is collaborative work with the Berlin Center for Advanced Therapies at Charité Hospital. And we've been working closely with this group and co-published a paper in Molecular Therapy Methods and Clinical Development, where we tested many different pharmacological interventions in primary T cells to find what gave us the best CAR knock-in rates. And here they're testing the HDR Enhancer version 2 in primary T cells, and we're able to considerably increase the rate of HDR to boost CAR knock-in at the track locus in primary T cells. So we saw about a 10-fold increase in the rate of CAR insertion with 4 different biological replicates here. And that's the graph on the right, with the blue bars representing a measurement taken at day 4 and the red being measurements at day 9. And then on the left-hand side of the graph in the corner at the bottom, we also looked at cell cycle analysis and measured the fraction of viable cells at different steps in the cell cycle and compared cells that were untreated and cells that were treated with the HDR enhancer and found no significant differences in the viability of these cells at different cell cycles. So this is a good demonstration of the use and utility of HDR Enhancer version 2 that enables high efficiency CAR knock-in with very little effect on the cells themselves. And finally, some conclusions on how to improve knock-in rates in primary T cells. So start with optimizing Cas9 and the delivery parameters to ensure good double-strand break formation and maximize the potential for HDR to occur. Next, consider the donor format carefully based on which attributes are most important for your work. For us, that was the HDR donor block for its high efficiency, low toxicity, and low risk of off-target integration. Next, you can improve the rate of donor insertion by including PAM and TRAC mutations to prevent cleavage by Cas9 after insertion by HDR. And lastly, the small molecule enhancer can greatly improve the rate of HDR, allowing for improved knock-in efficiency of 2 kb inserts like CAR. And so to wrap up, I just want to thank everyone on the IDT CRISPR team, everyone who contributed to this body of work, including our collaborators at Charité. Thank you all very much for listening and for giving me some of your time today. Okay, and I believe we're ready to take some questions. I do want to mention that if you're interested in seeing more of IDT's HDR products, looking for more resources, or if you want to check out our HDR design tool, you can visit our website, idtdna.com/HDR. Will take you to the HDR landing page, and there's a lot of good HDR material there. And if you have any specific questions that you want to have answered, feel free to email us in our general CRISPR inquiries email. So that's CRISPR@idtdna.com. And let's go ahead and take some questions.
Thank you, Bernice, for your very informative presentation. We received several questions from our audience. First question is, why do you see differences in editing between different primary T cell donors?
Yes, this is something that we're trying to figure out ourselves. It's likely to be something about DNA accessibility or chromatin structure that varies human to human. And all the data I showed in this webinar was analyzed by NGS and was background corrected for SNPs. So it's likely to be something sequence independent and more physically variable from donor to donor, but we're still investigating.
Okay, thanks, Bernice. Here's another question. Someone is asking, why do you think the activation method matters for CRISPR editing?
T cells need to be activated to be metabolically active. So it matters more for repair processes like HDR that require a lot of machinery recruitment. And there's evidence in the literature that different activation methods can be optimal for NHEJ versus HDR, and things like the source of the cells and other biological factors can make a difference in what's the optimal activation method. So it can impact CRISPR editing, and it's likely that you'd need to optimize in your specific experimental setup to find what is optimal for CRISPR editing there.
Great, thank you. Okay, so here's another one. Someone is asking about editing in primary T cells with Cas9 mRNA or plasmid. Is that something that you've tested?
It's certainly doable. We haven't tested it in primary T specifically. We've done all of our optimization with RNP delivery, but there is some web material on IDT's website that can make suggestions on how to use Cas9 mRNA and Cas9 plasmid.
Great. Okay. Oh, here's another question. So someone is asking, when should I use a donor oligo versus a donor block?
Good question. The donor oligos are synthesized as ultramers, so those can go up to 200 nucleotide total. So that's including the homology arms and the length of your insert. So generally for everything larger than 200 bases, use a donor block. Anything less than 200 bases, use a donor oligo. And that will also depend on your insert size, whether you need to compensate with larger homology arms. So it's more based on the total length that would make the decision of donor oligo versus block.
Great. And kind of in a similar vein, how do modifications on HDR donors increase HDR rates?
A couple factors here. The modifications protect against nuclease degradation in the cell. So that gives time for the donor to reach the nucleus and start the HDR process. And then for the donor blocks, those modifications prevent non-homologous integration or blunt integration at on and off target sites. So then more of your DNA donors preserved for that perfect HDR repair.
Great. Thank you so much, Bernice. Unfortunately, we've run out of time to answer additional questions, so we'll contact you directly if you asked a question and if it wasn't answered here today. Again, I would like to thank Bernice for her time with us today. Lastly, thank you again for attending, and we wish you the best of success in your research.